Hi, everyone. Let's just jump right into chapter 4. We're going to be talking about bacteria and archaea, introducing you to, their shapes and, their parts and everything like that. Here are our learning objectives for this entire chapter. Again, talking about the structure of bacteria, their shapes, how they're arranged, their structures that allow them to get around and move around. And how we would divide them based on maybe their cell wall structures and stuff like that. So bacteria and archaea versus eukaryotes. So bacteria and archaea, again, these are going to be our eukaryotes. So they do not have a true nucleus and they do not have any true membrane bound organelles at all, actually. So eukaryotes do have both of these things. And that will be what we'll talk about in the next chapter. Next chapter is about eukaryotes. And that's like you and I. So bacteria and archaea, on the outside of these organisms, we can have appendages and surface layers. Our appendages you would see here could be flagella, pili, the fimbriae, and nanotubes and things like that that allow transfer between cells. And then surface layers like a glycocalyx, which is a sugar layer. And then next that we have the boundaries. That's going to be our cell wall, cytoplasmic membrane, and sometimes an outer membrane, depending on the organism that we're talking about. And then inside, we have all these structures I'm not going to name for you, but we are going to talk about a lot of these structures and their function in the cells. This is a little bit more detailed picture of what a bacterial cell looks like. Since we're going to be talking about a lot of these structures in this chapter as we move through it, I am not going to be going over the details on this figure right now, but I would refer back to this figure if I were you, as far as, being able to find where these structures are and what these structures do and everything like that, so that you can, when you go back to study, I'm saying. Okay. So bacteria are single-celled organisms. They can reproduce, from one another. They don't need to reproduce sexually. So one cell can become two cells, and that's how they divide. They undergo metabolism just like we would, so breaking down molecules or building up molecules, as well as other processing of various nutrients that they might get from their diet and environment, right? So bacteria can act as a group. That would be forming colonies. So cells dividing and building up on one another in formation of a colony. They can actually signal with one another, especially if we're talking about biofilms, to let each other know how the biofilm is doing overall, so they can, decide if they want to jump ship or not, basically. Basically, there was a whole description on the sizes of the bacteria, but I don't need you guys to know that. So what I do need you to know is that there's a wide variation in the sizes of bacterial cells. Some of them can be quite small. Some bacteria are so small they can be smaller than the largest virus. So that's pretty impressive, actually. The three general shapes of bacteria: the coccus is the round spherical shape, the bacillus is the rod, the curved ones are referred to as vibrio. There's also some of the comma shaped ones are the vibrio. Spirillum is referring to a spiral-like shape. Corkscrew is exactly what it sounds like. The spirochete are super tightly wound coils. So, depending on what sort of curvature you have going on there, you can get more specific with those words. So coccus, bacillus, and curved. All right, pleomorphism is referring to the fact that you can have variations in the size and shape among cells of a single species. So when we talk about some of these bacteria, some of the individual cells that are associated with that kind of bacteria, like if we're talking about staph, so that'd be like Staphylococcus aureus, maybe Staphylococcus aureus would be able to have different sizes of cell shape or arrangement. So, but yeah, that's not the case with staph. That's why it's named what it is, which we'll talk about in one of the next figures. But that's just an example of how it could work if that were the case. So we have these different bacterial shapes and arrangements. And again, a lot of us might be used to hearing about, like if I talk about a bacterial cell being a rod shape, just thinking of that typical like rod kind of pill shape. But you can see here that there's a lot of variation within that. It could have a little bit of wiggliness or squiggliness associated with it. Coccus is just referring to general roundness in shape. It doesn't have to be perfect, but a lot of times when you look at something like Staphylococcus aureus, which is the one on top up here, that you can see that these guys clearly are very spherical in shape, as are streptococcus, which are cocci, round cells that are forming into a strepto means, or chain. That's another way of referring to that, but I always remember it as being like strip because that reminds me of the word. Anyways, are clearly round cells, right? So they're just stuck end to end to one another. And then we have the examples of the rods and the curved ones. So the Vibrio is the comma. Campylobacter has a spiral shape to it, etc. There's different ways that the cells can be arranged as well. So not just the shape of the cell, which could be a coccus for a round cell, or bacillus for a rod cell or curved. You can also have arrangements of those cells. So if they occur in pairs, like just stuck to one another. If you had a pair of cells that kind of look like this, that's a diplococcus. So this is talking about arrangements of cocci. Specifically, these are the round cells. So you can have them shaped up in stacked up in fours. So it's tetrad. Staphylococci. These are going to be irregular clusters. It sort of is literally the word referring to grape-like clusters. They're just irregular clusters, so clustered up together. Streptococci, these are the chains or strips, as I like to say, to help me remember. And then sarcina is a cubical packet. So if we had these guys here and then stack them up next to each other, for example, that would be cube, that'd be sarcina. So it doesn't have to just be, that size, it could be larger stackings. The other bacterial arrangements, maybe we're going to talk about the arrangements of the bacilli, which are our rods. They can be diplobacilli that are pairs with their ends attached, like so. They can be streptobacilli, so there should be chains just like that. So that would just be continuing on. Then we have palisades, where the cells of a chain remain partially attached and fold back, creating side-by-side row of cells. So these guys, the diplo, those guys that I had just drawn up at the top, could potentially still be attached at a certain part of it. And then we have, a folding up. And then we have this guy. Here's our attachment points. We'll just put this so you know where they're attached. Doesn't really matter where they're attached, but that's how these formations form. And essentially, they're gonna be like this, but they're just attached at certain points, and that's the palisade formation. So that's all really that I need you to worry about there. Biofilms, again, biofilms are whenever bacteria, so you'd have a part of a surface that has a roughness to it, bacteria come and attach to that rough, part, and they start growing and then start forming a layer. And then other bacteria can come and attach to that, and they can all signal with one another and let one another know how that piled up mess is doing, which is nuts, but they are able to do that. So that's a biofilm. They're microbial habitats. They have access to food, water, and atmosphere. This is what we're talking about with plaque on your teeth, right? So FYI, and this is how dental caries develops. You get cavities and stuff, breakdown of your teeth. Yeah, so that we can have secreting substances to attract other microbes or other signals that could tell, the other microbes that they, aren't having a good time. Maybe we should detach, for example, and go and live our own little lives. A lot of times the attaching has to deal with a sticky layer on the outside of the cells. That is what we call a glycocalyx. We'll get into the details of that momentarily. So that's how biofilms work. Bacteria can also have appendages. The two major groups are going to be the ones that provide motility and the ones that are going to be for attachment or channels. So the ones that are involved in motility are going to be flagella and axial filaments. Axial filaments are actually inside of the cell, like a spiral within the cell, and it can allow the cell to twist and move around that way. We often see these with the spiral shaped cells. I don't know why those words are tripping me up. So this is a picture of a flagellum. So the flagella is plural. Singular equals flagellum. And these will turn in a circular motion, rotate. Okay, so they will rotate. They do not whip around. They look like they should be able to whip around, but they do not do that. They will be rotating, and it works sort of like a motor through the layers of the cell envelope, so the total outer covering of the cell. And it does require energy in order for this to work, but that's the general structure of it. So it looks and operates quite a bit like a motor. We can have different arrangements of flagella on the outside of the cells. Flagella, again, are for movement. Okay, so that's why they're moving around and rotating that way. That allows the cell to propel itself and move itself around, in a appropriate environment. So polar flagella, we have flagella that are attached at one or both ends of the cell. So polar at one or both ends. So monotrichous, we have a single flagellum that has to be at one end or the other. Lophotrichous, we have a bunch or a tuft. So you can see here that this would be referred to as a lophotrichous. Amphitrichous, if you had this mess at both ends, then that would be amphitrichous. Again, remember, amphi means both. And then peritrichous, this is going to be flagella dispersed randomly over the entire surface of the cell. So just kind of all over the place. And you can see this guy here just coated in a bunch of flagella. So those are different ways that the flagella can be arranged. So they can be polar arrangements or they can be peritrichous. The polar arrangements include monotrichous, lophotrichous, a tuft, and amphitrichous, both ends. So if you can remember that amphitrichous, there's three words. Mono is one, amphi is both, like amphibians, right? And then lopho will be the tuft one. Chemotaxis refers to movement in response to chemical signals. So seeing that there's food, getting a signal from your environment that there's something favorable to the cell allows the cell to start moving its flagella in a way that will allow it to move closer towards that thing. There's also negative chemotaxis. That means that it will be moving away from like a poison or toxin or something harmful. If they're moving in response to light, then they would have phototaxis. Some of these bacteria, keep in mind, some of these bacteria are actually photosynthetic. Whenever we're using our flagella to move around, we move using the run and the tumble. The run is the counterclockwise movement of the flagella in a smooth linear direction. A tumble is we're going to be reversing the direction of the movement of the flagella suddenly. That will lead to a stop and a sudden change in course. So often you will go along, and then you will tumble suddenly, and then it'll be like, and then you'll start on whatever way that you turned or moved or whatever. So that could shift your bacterial cell completely around or whatever. It just depends on how much you moved on that tumble. And so that's how the bacteria guide themselves. And they move in a general direction. It's not so much a straight motion. But yeah, so in general, this first figure here is showing how if you have a flagellum, how you would move if you're just chilling and just, living in your environment and letting your environment kind of speak to you, but you're not getting pulled one direction or pushed any other direction. So they just kind of move around in random runs and tumbles, and they don't have any sort of direction. But as soon as we put in something that is an attractant, some sort of food or something that's drawing the organism to that area, then now we have movement towards that attractant based on our runs and our tumbles and how the bacteria are able to control and utilize those. Again, the axial filament is like an internal flagellum that kind of squeezes and turns and allows twisting and flexing of the cell in order to allow it to move that way. It's not as efficient, of course, as swimming with a flagellum. The other appendages, which we use for attachment or transfer or channel formation. The pilus, that provides adhesion, not locomotion. The fimbriae, adhesion, but not locomotion. The flagella can be used for attachment. It is mostly used for locomotion. And then we have nanowires. These are going to create a channel. So we do not use, we would adhere to cells, but we do not adhere to surfaces with these nanowires or nanotubes. start with the fimbriae. These are bristle-like fibers that are sticking out that allow for attachment to surfaces. This is usually used by organisms that are starting the formation of biofilms. And E. coli uses this to adhere to epithelial cells so that it can start an infection in a host as a pathogen. The pili, long, rigid tubular structures made of pilin protein. That's the protein that makes it up. This is found only in gram-negative bacteria. I would remember that. Gram-negative bacteria have the pilus. These are used in conjugation, which is kind of what it sounds like. It sounds like, that's what we call people having sex, right? So conjugation is sort of like that. So we're transferring DNA from one cell to another. That's going to be happening through those little tubes there. That's kind of what they're used for, and we can also use them for attachment in general. Whether or not an organism can make a pilus is determined by their genetics. So nanowires are thin, long tubes where we can transfer amino acids or electrons or small things like that. So nutrients, that sort of a thing. So those are nanotubes, nanowires. The S layer is a copy of single proteins linked together. It's just a layer on the outside of the cell that provides some protection in hostile environments. It's just, it allows the bacterium to survive harsh conditions. And then we have the glycocalyx. This is a repeating polysaccharide, so sugars repeating, so sticky, like sugar is sticky. It still is, even in this form, okay? So sometimes there may be a protein involved with the sugar, but not always. This, if we have it formed loosely on the outside of the cell and kind of gooey loose layer, it protects the cell from losing water and nutrients. This is called the slime layer. If it is a more tightly packed layer and it's denser and will allow sticking to things, that would be the capsule layer. So these are the pictures depicting what I was just describing to you for that, for the glycocalyx. I want to keep in mind that the slime layer and the capsule are types of glycocalyx. This is a picture of a capsule here. So you can see in the white, so the stain here, that negative stain is in the background, right? We can remember about this. That is that bluish color in the background. And then we have the cell stained in the middle, which is that purple little rod in the middle of that. And the white around it is the capsule. It couldn't stain. Capsules themselves do not stain. So that whole area represents the capsule that is surrounding the cell. Pretty neat stuff. We're going to be doing staining like that in the laboratory. Capsules in general are going to be formed by pathogenic bacteria as well as biofilm forming bacteria, and a lot of those can be pathogenic themselves. Capsules help protect bacteria against phagocytic white blood cells. Phagocytes are our white blood cells that eat up the bad guys, like bacteria that aren't supposed to be in our body. Phago means eating, cytic, cell related. So cells that eat other things outside of them, like other cells, like bacteria. So this helps those bacteria evade that sort of a situation and protects them from us getting rid of them with our immune system. Then we have biofilms like the plaque on our teeth. And this is just, they'll build up and stick to one another using those glycocalyx layers. That they can also be associated with, like they're saying, long-term indwelling artificial devices like a pacemaker or something like that. All right, the cell envelope is referring to the entire outer, coating of the bacterium. So this is going to consist of a cell wall and a cytoplasmic membrane for sure. Some bacteria have an outer membrane. There are some bacteria that do not have this. This is almost all bacteria. Okay, that's a word that means almost all, apparently. I don't know where I got that from, but okay, so that whole thing acts as the protective unit and helps hold everything into the cell. The gram stain was the stain that was developed to kind of look at that cell envelope and differentiate cells based on their cell envelope. Gram positives, they have a thick cell wall. Gram negatives, I'm going to show a picture of it, so don't worry. Gram negatives have a thin cell wall. Both of them have inner cytoplasmic membrane. That's just the membrane on the inside. So it looks like this. So here is your cell. Here is your inner membrane, your cytoplasmic membrane. Here is your cell wall. And I am, sorry, I clicked on a thing. I'm just trying to make it look like a thick layer. Okay, so I'm not going to go all the way around, but you get the idea. Okay, and then you could have maybe an outer membrane around that. That would make you gram negative. Or if you're gram positive, you have a really thick cell wall on the outside. This is like a super exaggeration, but that would be gram positive. So that's the difference between these. So this is showing a picture of that. Here on the right is what I'm going to start with, actually, which is our gram negative. You can see this is a lipid bilayer, just like this is a lipid bilayer. And in the middle in here, we have the peptidoglycan, which is what that molecule is making that up. We have a thin layer of that, very thin layer. Yeah, so there's that. And you would see that the picture had depicts like a different color on the outside of this, like almost like these are different kinds of lipids than what we normally see in the normal membrane. That's because they are different. That's important to remember. Yeah, because they're going to associate with lipopolysaccharides, and we'll get into that in a moment. I don't want to talk too much about it, but this is the cell wall, the thick cell wall. So this little thin layer in here, that's the cell wall made of peptidoglycan of the gram negative bacteria. This thick buddy over here, that's in the gram positive. So you can see the big difference there. Gram positive having that thick layer for their cell wall. So the cell wall itself, that thick layer, that peptidoglycan layer, helps determine the shape of the bacterium and how well it can sustain its shape when exposed to too much or too little solute in the solution that it's swimming in, right? So that's osmotic pressure. So it won't burst or collapse due to that change in salt concentration surrounding the cell. Peptidoglycan itself is glycan chains, so sugar-based chains, and those are linked across one another like a big scaffolding with short peptide fragments. Now, wouldn't you know it, that peptide fragments and the glycan chains, that's where the word peptido, and then glycan. So that's why it's called peptidoglycan. It's not meant to be some sort of secret fancy new word. It really isn't. So that creates a very strong but flexible framework, like a big scaffolding. So here's what it kind of looks like. And I don't need you guys to know too much detail about this, except that these guys here. So here is our, these are our sugars. That's why they're drawn like sugars. Remember, this is what sugars look like whenever you draw them. These are our sugars. And these guys are linked together by little bridges. These are our peptide crosslinks, these green lines in between. And the sugars are going to be N-acetylglucosamine, which is NAG for short, and N-acetylmuramic acid, or N-acetylmuramic acid, NAM. So they're alternating. So that's why it says G and M. That's just the last letter of that. Okay. So G and M and G and M and G and M. And then we're linking across crosswise with some peptides. Peptides are just strings of amino acids. Don't freak out over that. So our gram positive cell wall, we have those stacked up really, really high, like a high, thick scaffolding. Okay. It's very thick, 20 to 80 nanometers thick. We have incorporating within that teichoic and lipoteichoic acid. What you need to know about these guys is, number one, they're associated only with gram positive, with the thick gram positive cell wall. They're involved in the cell wall maintenance whenever the cell is undergoing division to make a new cell. These guys are involved in that to maintain the structure of the cell wall around the new cells. They also give an acidic charge on the surface, so negative charge. So with the gram negative cell wall, we have a very thin sheet of the peptidoglycan. It's only 1 to 3 nanometers thick. It is a little bit rigid. but it is very thin, so it's a little bit more flexible. But so that therefore it's great. So it's greater flexibility, which is nice for it, but it's also more sensitive to lysis. So being burst open. So it doesn't have that structure there that the thick ones had whenever they had the thick wall. So interesting, if I tell you, we've already mentioned previously that Staphylococcus, like Staphylococcus aureus, because it can grow on skin, right? That's a salty environment. They are gram positive. So they have that thick cell wall that's helping protect them. Whereas some bacteria that are gram negative, that have like E. coli, they aren't going to do well living on the skin because they don't have the structure in their cell wall. It's too thin of a cell wall. It's not going to protect them from lysis whenever we're being exposed to all that salt on the skin. So interesting. There you go. You learned a thing. Probably don't care about it, but there it is. Right. So non-typical guys, the guys that are weird from the norm. We have the acid-fast bacteria. That includes Mycobacterium and Nocardia species. I really want you guys to focus mostly on Mycobacterium. Okay. They contain a mycolic acid, which is also known as cord factor. It's essentially a wax that makes them resistant to being killed with certain chemicals and stuff. And we use the acid-fast stain in order to identify organisms that possess that cord factor, that mycolic acid. And that's why these are called Mycobacterium, mycolic acid. And that waxy substance, so that specific way of staining and the molecule that we use for the staining, that is going to associate with that waxy mycolic acid and not with regular cells. So that's how it stains pink with that carbonyl fusion. And it rinses out from, carbonyl fusion rinses out from normal cells. Then we have the mycoplasmas. These guys don't have any cell wall at all. So their membrane is going to be stabilized by sterols, kind of like ours. Ours is not the exact same, but very similar. They have, because they don't have that structure, the rigidity involved, they don't have a like typical shape. So they're very pleomorphic. So they can be all sorts of different shapes. Pleomorphic means they don't have a set shape of the cell. Okay. The cytoplasmic membrane. This is the one that all of the bacteria have, whether they have, outer membrane or not. That would be determined based on gram negatives have the outer membrane, whether or not they have the actual cell wall, whether it's thick or thin, or mycoplasma not having one. Then everybody has the cytoplasmic membrane, and this is just the membrane that is containing everything within the cell. There's a lot more going on there than just that. So in mycoplasmas, they have a lot of sterols there to give a little bit more stability and rigidity in that, because they need that because they don't have a cell wall. Archaea are kind of different completely. So they have branched hydrocarbons instead of fatty acids, which we were just talking about fatty acids, right? About phospholipids and how they have the charge and they have the fatty acid tail, and how that's what gives that whole lipid bilayer its structure. So this is just one side of the lipid bilayer, but yeah, so we have a whole bunch of membranes, a whole bunch of proteins dotted all over the membrane for it to perform whatever functions that the cell needs performed along the membrane. We have a lot going on inside of a bacterial cell associated with the cytoplasmic membrane. We have organelles, so there's little structures within our cells that perform functions for us. We have organelles that do a lot of this work for us, whereas bacteria have to do this all associated with their actual inner membrane. So this is where they have their energy reactions, their nutrient processing, making new molecules to build up, parts of the cell. And then it's also where they regulate all the transport of passage of nutrients into the cell and waste out of the cell. Membrane itself has something called selective permeability. So water and little small polar molecules, small uncharged molecules will diffuse freely across that membrane because of the unique ability of water to get in between those charged heads in there. And it's a small enough molecule, and other small molecules can get in there as well and can cross the membrane. So that's selective permeability. Only certain things can get across. Other things can't get across without some kind of help in some way. Okay, the gram-negative outer membrane. So again, only the gram-negatives are going to have the outer membrane. This is very important, incredibly important actually, because on that outer membrane, they have something called a lipopolysaccharide. Lipo being a fatty acid associated thing, right? And then like a lipid. And then polysaccharide is sugars, right? So this is very important. This is dealing with endotoxin. This stimulates fever and shock. Whenever we are talking about our reactions to having like septicemia, where you have bacteria flourishing in your bloodstream, endotoxin is part of what can have you have a shock reaction to that sort of an infection. So that is going to come up over and over and over again. And I need you guys to be aware that endotoxin is associated with gram-negative bacteria. All right, sorry, I had to pause it for a second. So endotoxin is associated with gram-negative bacteria. Lipoproteins, again, that's going to be lipids like fats. Associated with proteins, they're going to anchor the outer membrane to the peptidoglycan layer so that it stays on to that peptidoglycan layer and doesn't just disassociate randomly when there's some sort of damage or something like that. So that's what keeps that stuck to it, is those lipoproteins. All right, porin proteins. These are proteins that are involved in the gram-negative outer membrane that span the outer membrane and allow small molecules to penetrate little pores, essentially, okay? So the size can be altered to block the entrance of harmful chemicals so that if the bacteria is experiencing some sort of damage from harmful chemicals, it can close off those pores, those porin pores. To defend against whatever that toxic substance is, including antibiotics. So the genes that encode for the ability to manage this whole system is something that can be associated with antibiotic resistance. Okay, the gram stain is the stain that is developed in order for us to see the difference between two bacteria based on their outer, envelope, their cell envelope. This is how it works. All right. At any given time, this is going to be showing you what color the gram-positive and the gram-negative cell is during the staining process. All right, that peptidoglycan, we're going to add our gram. We're going to make our heat-fixed slide first. You guys remember about this. Yes, heat-fixed slide to get our cells stuck into place, to prepare them for staining, to keep all the structures in place. And to kill the bacteria themselves. So we have our thick peptidoglycan layer here and our thin cell wall in the middle for the gram-negative, right? So we're going to be adding our first stain, that is the crystal violet, and that's going to get stuck inside of that lattice of the gram-positives, okay? Then we will treat with iodine. Iodine is something called a mordant, which just helps fix the crystal violet into place if it is actually getting, trapped there properly. So that just helps trap it in there and makes a complex so that it would not be able to come out. So then we rinse with alcohol, and if it didn't get stuck by the mordant, the crystal violet will rinse out with the alcohol. So that alcohol is also going to break down that outer, so the alcohol breaks down that outer membrane of the gram-negative. So that causes the little bits of crystal violet that did happen to get kind of meshed in there, causes them to be able to be very well rinsed out with the alcohol. And it's just alcohol, just ethanol is what we would be using at this step. And then after we have removed that with a water rinse, then we will apply a counter stain, because at this point, our cells would be clear and you wouldn't be able to see them very well in the microscope if they were gram-negative, because we just removed all the crystal violet. It's not purple anymore. So our gram-positives remain purple because we locked the crystal violet in place with the iodine. The purple is quite dark. You're not going to see the safranin, which is a pinkish dye. So that red, I say it's red because it helps me remember which one's which, but that red dye will stain that colorless cell. So then we will have a red cell that when we look at it. So gram-positive cells are purple, gram-negative cells are red. So gram-negative bacteria, that outer membrane can make them resistant to some antimicrobial chemicals. And it makes them more difficult to inhibit or kill their growth. And then, yeah. So we treat them differently based on their outer envelope, that cell envelope. And you guys probably don't think about that very much, but that is the case when you're treating something medically with a medication. We have to consider whether or not it is gram-positive or gram-negative. If it is a bacteria, that changes the protocol quite significantly, depending on the medication that you could be using. So it's incredibly important for that. The cytoplasm is the goo inside the cell. What do you want me to say? It's mostly water. Of course it is. This is where a lot of the biosynthesis of bacterial cells is going to be happening. So we have everything that is associated with a cell that isn't actively working at the membrane to like make energy or something. It's all going on in the cytoplasm. The bacterial chromosome is a single circular strand of DNA. And it's going to like congregate or aggregate, I guess is the word they're using, in an area called the nucleoid region of the cell. So, we have other stuff going on in the cell at the same time. It's not all over the place. So it's this little area called the nucleoid region. The DNA is quite tightly coiled around protein molecules so that it can fit into that area and not be, in the way of everything else going on in the cell. Plasmids are non-essential pieces of DNA. These are separate double-stranded circles of DNA, much smaller than the chromosome typically. Okay, small little circles of DNA, small. I know people get confused over that for some reason. I don't know why. They're not the same size as the chromosome. They are circular, yes, but the pictures, I guess maybe that's what makes people confused, but no. These are small. These guys are small. They only have a few genes, okay? Usually like 3 or something. These can be duplicated and passed on between bacteria, can confer protective traits like the ability to break down an antibiotic, for example, so antibiotic resistance. And we can even put our own genes into plasmids and control when they are or are not expressed by bacteria in order to force the bacteria to produce whatever protein we want them to. A ribosome, so they are made of ribosomal RNA and protein, a little bit of protein. They are made up of subunits, a small and a large subunit, and they work together to basically create the machinery that is involved in translation, which is making proteins from reading messenger RNA. Bacterial ribosomes have a 70S small subunit, and the eukaryotes have 80S small subunit. These are just sizes of those small parts of the ribosome. I don't need to worry about that. Don't worry about that. Don't worry about that, by the way. Okay, inclusion bodies. These are just sites where we're going to be storing things. Cytoskeleton. This is the internal structure of the cell. So this is how we're moving things along inside of the cell as well to give shape and allows for movement within the cell of certain molecules. And if we can target this with certain antibiotics, maybe break down the ability of the cell to maintain its structure and ability to move things within. Endospores, these are those guys that can help the bacterial cells withstand hostile conditions. So they exist in 2 forms. They're either a vegetative cell, which is the normal metabolically active state, and then the endospore version, which is inert. It's resting. It's waiting for conditions to improve. So this is in harsh conditions. Sporulation is whenever we are forming those endospores. So that's the whole process involved in forming an endospore. That's referred to as sporulation. Spores are very resistant to a lot of things, including heat, drying, freezing, radiation, and chemicals. A lot of chemicals in radiation. So it's very surprising the things that they can withstand. So that's why we have to be very thorough if we want to get rid of these spores that are associated with certain diseases and not have them be spreading everywhere, especially in a hospital setting. So this is, we might maybe have less nutrients than what we need in order to survive. Carbon and nitrogen sources are usually the main cause for forming an endospore. The sporangium is the sporulating cell. So that is the cell that decides that it's going to make an endospore. That's the sporangium. It takes about 6 to 8 hours to make an endospore. And again, it's just to protect the genetic information of that cell. So we have a normal cell here. Step 1, normal cell. Step 2, step 2, we'll start breaking the cell into parts to separate the chromosome, and maybe we doubled our chromosome so that we have part of it being shoved into the endospore compartment, essentially. So that's the forespore. And this part is the part that is making the spore. That is the sporangium. Anyways, then we start sort of walling it off, and then we encircle it with our little layer here. And then we start putting a cortex layer. It's our covering or protective covering on here. And then it's going to mature. And then we have our endospore covered in its cortex, protecting its little genome in its core, waiting for conditions to be favorable again. And then when they are, we can break out of our cortex and become a vegetative cell again. So that's basically what's going on there, the steps that are involved in sporulation. So sporulation is forming the spore. So that's everything up to here. If we are a spore and we are becoming a vegetative cell again, so this process here, breaking out and becoming vegetative again, to rejoin society, if you will, that part is called germination. The whole rest of this outside part is called sporulation. So germination, that's going to happen whenever we have favorable conditions. Again, we have water and whatever the germination agent is, so carbon-containing nutrient or something. The germination will stimulate the germination agent will stimulate formation of hydrolytic enzymes. So just breaking down the covering, the cortex, and then we will rehydrate and become a vegetative cell. A vegetative cell, that part process, the germination process, that only takes about 1.5 hours. So you come into contact with anthrax spores, tetanus spores, gas gangrene spores, or botulism spores. It only takes those guys about an hour and a half to go from spore to become, dangerous pathogen. That's all it takes. So these spores always going to be associated with bacillus. Endospores are going to be associated with bacillus and Clostridium species. In this case, these are just examples of disease-causing versions of these guys. And there's a lot of them out there, so I'm just saying. So Clostridium species and bacillus species are the biggies. They are rods, by the way. Right, so they're an issue in hospitals where we want things to be sterile because we need to have a way to remove them, and sometimes it's really difficult to do that. We can't autoclave everything. So that's usually how we would sterilize something, but you can't do that with every single thing. So we have to have ways to get rid of endospores, and endospores will be resistant to normal cleaning methods. Boiling water doesn't get rid of them. Soaps and disinfectants, a lot of them that are used in hospital settings will not get rid of them either. So these guys also tend to stick around in laboratories and will contaminate cultures and media and get spread around all in a laboratory because, they're not getting removed by disinfectants. Then hospitals have to have some way to, protect against the spread of these sorts of things. So, and obviously we need to control it in a canning industry. The damaged cans often associated with the gas formation of the Clostridium botulinum bacterium. So if you do see a sort of swollen can, if it's swollen at all, or if you open it and it hisses, maybe don't eat it. That's probably got botulism in it, so don't eat it. That's a very severe toxin. Don't mess around with it. I don't care what you do as far as like injecting it in, for beauty purposes, that's a different situation, but ingesting it, don't just look at a food and be like, oh, who cares? People get this toxin all the time and injected in their face. Who cares if I eat a little bit of it? No, your body cares. So don't do it. So archaea look a lot like bacteria. if you looked at them under the microscope, you wouldn't be able to really to know the difference, or not really. But their genes, their genetic makeup are more similar to us than they are to the bacteria. So that creeps me out a little bit, but that is the truth. Yeah, I don't know. They're also just completely different, of course, than anything else. They've got different cell wall makeup, but they've got different, membranes going on and all that. They do not have true peptidoglycan and cell walls, so it's an unusual cell wall. They live in habitats that are extreme, so high salt content, very high temperatures or very low temperatures, acidic pHs or very basic pHs, just at the extremes of all of these, right? And they also tend to be methane producers. They are the hyperthermophiles, the extreme halophiles, and the sulfur reducers. So these guys are our extreme environment living organisms. So what are methanogens? These are guys that convert carbon dioxide and hydrogen gas into methane gas. This is often going to take place in anaerobic swamps. So there's not oxygen available in the water of these swamps really, and down in the bottom of lakes and stuff. So we have methane buildup sometimes in these areas, and that is what people talk about sometimes with the methane, bubbles coming up. But see, it's these guys, it's our archaea. We think that this process may contribute to global warming, so it's not just people doing it. It's mostly people though, guys, come on, let's be real. All right, so the extreme halophiles, these are the guys that are going, so these, again, we're talking mostly about archaea here. They require salt to grow. They don't just grow well in salt. They don't just grow fine in salt. They have to have it in an environment. Without it, they can actually die because they are so well adapted to having the salt around them all the time. So they are going to be in inland seas with high salt content, salt lakes with high salt content, like the Dead Sea. That's an inland sea, by the way. I don't know why I called that a lake just now, but you guys get the idea here. Salt mines, salted fish, all of that stuff. These guys can grow in those sorts of environments. So they use a red pigment to synthesize energy in the presence of light. So you can see like large, like swaths of like salt, like pond, river, not river really, lake sort of area where it's all red. That's the archaea. That's exactly what's going on there. So we have the psychrophiles and the hyperthermophiles. The psychrophiles, these are going to grow at very low temperatures, so like freezing temperatures. And then the hyperthermophiles grow at very high temperatures. So they will flourish at temperatures between 80 and 113 degrees Celsius. I want to remind you guys that 100 degrees Celsius is boiling. So they flourish at these temperatures, and they cannot grow at 50 degrees Celsius or below. That's too cold for them. Our body temperature is 37, by the way. And so yeah, these are the guys that live in the vents, the submarine vents with like the hot water coming out of the vents and all that, and in volcanic waters. They can also usually, a lot of these guys are cross tolerant, right? So if it can tolerate the heat, it often can tolerate salt and acid as well, and vice versa, just depending on what organism you're talking about. But yeah, so that's archaea. Yeah. Right, so we have classification systems in place in order for us to classify the different bacteria and the archaea, and then separate them even from one another, and different ways that we would approach that sort of a thing. We did talk about this a little bit in the taxonomy area, right? So a lot of bacterial microbiologists, whenever they are characterizing these organisms, they are going to be talking about biochemistry of the organisms, their genetics, and their molecular traits. Nowadays, this is the modern society, right? Before, it was mostly based on their shape, their arrangements, their growth characteristics, where they came from, their habitat, what it took for them to grow it, to culture it, and what they actually preferred, and the color of the colonies, and all of that sort of stuff. So they would use that information because they didn't have the testing methods that we have today with our modern technology, the biochemistry, the genetic, and the molecular traits. What do we mean by molecular traits? That's like actually testing like the lipid content of these organisms, for example. So not the genetic necessarily, that's the genetic is genetic, right? So evolutionary relatedness is determined through comparison of ribosomal RNA. So this is freaking small subunits, right? So how related are these organisms based on everything has to make a protein? We need ribosomes in order to do that. So we use the genes that kind of code for those ribosomes in order to compare how similar these organisms may be. So that is what we're talking about with the genetics and their relatedness and their ancestry based on that. So Bergey's manual, this is how these are like traits of these organisms that we would use to determine what they are that are already determined based on studies done by other people. So they compiled it together in this manual. It uses phenotypic traits, which is like what you would observe, right? So the color of the organism, the size of the organism, the shape of the organism, the colonies, and as well as biochemical traits. And we can also use genetic traits as well now. But yeah, so this is how we would determine what bacteria is what by doing these tests and then breaking it down. Oh, well, is this gram positive or gram negative based on this stain? It's gram positive. Okay, what next? Is it acid fast? Okay, what next? Is it, endospore forming or something like that? And we can break it down into what sort of organism we think it is based on that sort of testing, which is what you're going to be doing in the laboratory. So, Bergey's Manual of Systematic Bacteriology. This is dealing with phenotypic information like results of biochemical testing, along with ribosomal RNA sequencing. So genes are looked at here, as well as other phenotypic traits like they used in the olden days. Both. Determinative, this is only phenotypic. You don't have any genetic information, you might use Bergey's Manual of Determinative Bacteriology. Yes, you need to know the difference between these guys. So systematic, we had the inclusion of the ribosomal RNA genetic sequencing. We don't have that with determinative. It's only going to be phenotypic. So if you are studying for medical microbiology and you want to know about certain traits about these organisms that you are potentially dealing with in your patient, this might be the manual that you might consult for that because you don't need to know the genetic information for it. So we can classify bacteria and archaea based on their envelope, basically. Does your bacterium or archaea, whichever, does your organism have gram-negative cell walls? If it does, then it's a gracilicute. Does it have gram-positive cell walls? If it does, then it's a firmicute. Does it have no cell wall? Then it is a tenericute. And then if it is an archaea, so it has to be weird, it has a weird cell wall, that is the Mendozicute. Okay. Do what you need to in order to remember these guys. I always remember the firmicutes is firm. The gram-positives are the ones with the thick, strong peptidoglycan layer, the firm peptidoglycan layer. So they are the firmicutes. I had this boyfriend. He was my first boyfriend in middle school or something like that. And his name was Joey Mendoza. He was a nice kid. Anyways, that is a long time ago. That is, ancient. And to me, that's archaic, right? So that's how I remember. His name was Joey Mendoza. So it's the Mendozicutes. He was cute too. So there you go. Mendozicute. That's how I remember that. But as far as gracilicutes, if you can break down firmicute and Mendozicute, then just remember that gracilicute starts with a G, like gram-negative does, and then I'll give you gram-negative over the ones that have no cell wall. That leaves you with tenericute for that one, if you can remember the words. So I don't know, maybe that'll help you, maybe it won't, maybe that's just BS. So whenever you are trying to diagnose your patient, you might look at all these different sorts of aspects of the cells that you are, culturing from your patient. Gram-positive or gram-negative, or no cell wall, because the mycoplasma are associated with pneumonia. So you might actually deal with mycoplasma that do not have cell walls. So those are important to think about. What other subgroups you can divide it into based on cell shape, the arrangement of the cells. So cell shape, what were the cell shapes? You guys remember? That's gonna be the cocci, the bacilli, that's the rods, and then the curved. So it's like all the curved ones. So that's how you would break that down with the cell shape. The arrangements, so that's going to be like staphylo, strepto, the palisades, the diplo, all of those sorts of terms. Then we have based on whether or not they can use oxygen or if they're even going to die in oxygen and that sort of stuff. So the aerobic organisms use oxygen. The anaerobic organisms do not use oxygen, and a lot of them die in the presence of it. Then we have facultative anaerobes. Facultative anaerobes can use oxygen or survive without the oxygen. So then we can use that information to sort of break it down into certain categories. And I'm not going to go through everything on these slides, but these slides keep going on and on and on. So we start off with gram-positive, let's say. So we have gram-positive cells. We've stained our cells, they're gram-positive. Oh, they look like they're cockeye, but they're in chains. Cockeye, they're in chains. Sorry, these are supposed to be medically important organisms. So that might include Streptococcus species. So that would be like what causes strep throat, Streptococcus pyogenes. So this might help you address how to treat the organism or how to better grow or culture the organism if you're having issues with it. And what sort of antibiotics to choose and all of that, breaking it down this way and looking at it from, here's our gram-negative ones and breaking them down. Okay, well, are they aerobic or anaerobic? Okay, they're anaerobic. I was going to look at the anaerobic. There we go. Rods. That's what I want to look at. If they're anaerobic rods and, maybe it's Yersinia, which would be Yersinia pestis, which causes the plague or something like that. Like, I don't know, you just break it down based on their characteristics and be able to categorize them for that. And remember, if they have no cell walls, they're going to be the mycoplasma. And there's also ureaplasma, which are basically mycoplasma, but they're associated with urinary infection. So these are our medically important ones. If you guys want to go through these and look at these and look at their traits and, familiarize yourself with these, you will see these. A lot of these anyways. You might not see Yersinia pestis, but a lot of these guys you will see when you're working in a hospital, for those of you who are going into nursing or PA. All right, so a bacterial species is just a collection of bacterial cells that have an overall pattern or a similar trait. So, how do we determine that this is a bacterial species? This is how, right? So they all fall into Escherichia coli, but we can have subspecies, strains, or types. So those are bacteria that we say are in the same species, but they have certain characteristics that are defining to them. So this might include what we call with Salmonella enterica. And there's a lot of different ones of this. So they're all Salmonella enterica, but they could be typhi. They could be, so we would just call that Salmonella typhi, but they're not different species. I believe they're just different subspecies. Typhimurium, whatever. So there's a whole bunch of them. I'm not going to go through all of them, but you get the idea. They're still all the same species, but based on whether they or not they have a certain toxin or this specific trait, then we can put them into subspecies. So a serotype is maybe you have a species that have a distinct pattern of antibody response. So if you're exposed to it as a person with that illness, the antibody that you make against it is going to be different than the others that are in that same species. So that's a serotype. So that's how your body is responding to it. How antibodies that are made to fight off that infection respond differently to that serotype. Okay, so looking back on the whole chapter, here's a question for you. What are the differences between the bacterial chromosome and plasmid? And I will leave you guys with that and see you guys next time.